Hello, it's Miss Nicholas. I'm going to talk to you a little bit today about the pancreas and the role of the pancreas in regulating blood sugar. Excuse my voice, I'll go ahead and tell you that. Let me get that over with. I know I sound a little scratchy. I did some really loud screaming at the Alabama-Auburn game last weekend, and I'm still recovering from that. So anyway, we're going to go ahead and get started talking about the pancreas. The pancreas is an endocrine gland, and it is shaped somewhat like a tadpole. So if you look here on your PowerPoint, we're talking about, we have already started swapping slides. Let's back up here. It didn't like what I did there, did it? Get the pen, turn it blue, and take me back to my slide. There we go. So the pancreas is a tadpole-shaped organ. The tail end of the pancreas is located near the spleen, and the enlarged head end is encircled somewhat by a part of the small intestine. Do you remember what that part of the small intestine is called that's located closest to the stomach? The stomach empties into the duodenum or the duodenum. So this is that part of the small intestine here. Okay, so let's talk some more about the pancreas. The pancreas is composed of two different types of tissue, and they have two different types of function. There is an endocrine portion, but the biggest part of the pancreas is composed of exocrine cells. Do you know the difference in endocrine and exocrine? I know you do. You've talked about this before, surely. If not, you've read about it already. Exocrine glands secrete into a duct and then onto a surface or into a body cavity. The endocrine cells are going to secrete into the bloodstream. So you knew that already. I know you do. But I wanted to point that out, that we do have both types of tissue, and the biggest part of the pancreas does serve exocrine function. Those exocrine cells are called acini or acini cells, A-C-I-N-I. Okay, so they're actually, let me correct that, they're acinar cells. Okay, right here's that word. But the little clusters that they all group together in are called acini. So the acini are the clusters of acinar cells, and they produce digestive enzymes and digestive juices that are secreted into tiny little ducts that feed into a much larger pancreatic duct, and then the pancreatic duct empties into the duodenum. So this all helps with the digestive process. You'll learn more about that when we do the digestive system later on this semester. But I wanted to go ahead and give you just a little preview there to get you started on it. Now, remember I said the biggest part of the pancreas is composed of the exocrine cells. But what we're studying now are endocrine glands. So we want to talk about the endocrine cells. So the endocrine cells make up a very small percent, about 2% of the pancreas, and they're arranged in little clusters of cells that are known as the islets of Langerhans. Okay, so we're looking for these islets of cells that are going to be the endocrine hormone producing cells. They're arranged in little islets, and when you look at these on a slide, you'll see that they stain differently. So when you're looking at this particular picture, this is an islet. So all of these darker staining cells out here are your exocrine cells, and here within the circle we have the endocrine cells. Now there are different types of cells located within each of the islets. You have alpha cells that produce glucagon, beta cells that produce the insulin. So that's one of the ones we're going to talk about, or we'll actually talk about both of those. But there's a third type of cell as well, and those are the delta cells. So it's supposed to be a triangle. Okay, the delta cells secrete a hormone known as somatostatin. Somatostatin. Can you tell anything about that word by breaking it down? Well, something that is somatic means of the body, and statin, to remain static or the same. So somatostatin is a hormone that's secreted by the delta cells. You're going to hear that again when you're talking about another gland, but let's focus on this one now. The somatostatin releases the delta cells release the somatostatin, and what it does is to inhibit the release of insulin and also inhibit the release of growth hormone. Growth hormone should be pretty obvious. It's going to cause the body to grow, right? Cause structures to undergo mitosis, cells to undergo mitosis, and structures to grow. So somatostatin inhibits the release of growth hormone, so the body remains the same or no growth occurs. Okay, so let's move on to the next one. Let's talk about the first of these hormones. Now remember, we said that it is the alpha cells of the islets of Langerhans that produce the glucagon, and the glucagon is a hyperglycemic agent. What does hyper tell you? Hyper means above or over, and glycemic refers to sugar. So could you tell what the effects of glucagon are simply by that statement? Of course you can. Hyperglycemic. Its action is to raise blood sugar. Okay, so this is going to be released when blood sugar levels do what? When they drop, because remember its action is to raise blood sugar. Okay, so it's regulated by a humoral mechanism. Humoral should tell you something as well. It means that it's regulated by the levels or the concentrations of something in the blood. So when blood sugar levels decrease, blood glucose decreases, the pancreatic alpha cells are going to be stimulated to release glucagon. It's going to get into the bloodstream and travel to target cells, primarily in the liver. Now once it gets to the liver is composed of cells that are going to have several functions, and one of which is to store glucose. Now remember, glucose, if you've seen the structure of glucose before, it's a cyclic hexagon shape, or that's the way we draw the shape, and that's a monosaccharide, a single sugar molecule, right? Well, we need all the glucose we can get to make ATP so our body has fuel. Well, we use glucose in this shape, but we don't store it like this. What we do to store it is to hook glucose molecules together to form long chain molecules. So when we form those long chain molecules, we're going to have another one out here, and this is going to go on and on and on and on. So this long chain molecule of glucose is glycogen, G-L-Y-C-O-G-E-N. Okay, so we store glycogen, and the liver is where that occurs, and the liver and muscle cells as well. So the hepatocytes have stored the excess glucose by hooking them all together, forming glycogen. All right, so now blood sugar levels have fallen, and the pancreas alpha cells have released the glucagon. It's traveled through the bloodstream, reached the target cells, which are in the liver primarily, and it's going to cause this glycogen now to be broken down into glucose. Okay? So glycogenolysis. Lysis means to break down, to lyse, to cut. So glycogenolysis is going to occur, the breaking down of glycogen to glucose. The glucose molecules then enter the bloodstream, and blood sugar levels are going to increase. Now, also what happens is that the liver is going to be stimulated to form glucose from non-carbohydrates, from non-sugar molecules. And it's going to do that by breaking down amino acids and using components of those amino acids and breaking down fats. Remember, fats are composed of glycerol, glycogen, glucose, all related, right? So we can use that glycerol. Let me complete my sentence there first. The fatty acids and the glycerol make up the lipids, right? The fats. So when we break down those lipids, break down those fats, we're creating fatty acids and glycerol. And then the glycerol molecules can be used to make glucose, okay? So all of that has the effect of causing blood sugar levels to rise. So the liver is going to release stored glucose. It's going to break down glycogen into glucose and also form glucose from molecules that are not normally used to form glucose. But when glucose levels are low, we do access those others as well. So these mechanisms are going to cause enough blood glucose, or cause blood glucose levels to rise. And that ensures, of course, that you've got enough fuel for the body. So glucagon is released when you're sleeping, when you're exercising, or any time that you need more sugar to get into the body. All right, excuse me just a second. Let me grab some water. Okay, now let's talk about a hormone that you're probably all familiar with already, and that's insulin. Insulin is a peptide hormone. It's created by amino acids. It's synthesized by the beta cells of the pancreas. And when it's made as a precursor molecule that's actually known as pre-proinsulin. Okay, so it's got additional components. Now, in the endoplasmic reticulum, the ER, you remember that from your cell structure study in 201, right? The ER modifies things and changes things and helps them to become the functional shape that they need to be. So the first thing that happens after the beta cells are making this pre-proinsulin, the ER is going to chop off part of that molecule, and now we have proinsulin. So proinsulin is actually going to be stored in the vesicles in the cells until the body needs to release the insulin. All right, so the effect of insulin is to lower blood sugar levels. So it is hypoglycemic in action. So when we eat or we drink, our blood sugar levels, of course, are going to rise as the body's digesting whatever we've taken in. As it's digesting, there's going to be free amino acids and free fats and free sugars floating around. The sugar, of course, will be absorbed into the bloodstream. And that is going to stimulate the pancreas to release insulin. Now, once insulin has been released, it's going to travel through the bloodstream to target cells all throughout the body. So this one has a very wide range of target cells. Insulin is going to target almost every cell in the body. And it binds to extracellular receptors. Remember when we talked about intracellular receptors and extracellular receptors? If something binds to an extracellular receptor, that should tell you it's a non-steroid hormone, right? Which we knew that already because we talked about it at the beginning. Okay, so anyway, it binds an extracellular receptor and it causes glucose transporters to come from down in the cytoplasm of the cell to the cell membrane. Now, once those transporters are at the cell membrane, their job is to bring glucose into the cell. So all this glucose is coming into the cell and is going to be used to turn on the cellular machinery, which creates ATP, so cell respiration begins. So the cells of the body are making all this glucose, are making all this ATP from this free glucose. And let me go ahead, I see I've got a note here. I wanted to be sure and remind you that there are some cells of the body that don't rely on insulin binding to take glucose in, and those cells are found primarily in the brain, the liver, and the kidneys. So they're able to transport glucose into the cell even when insulin is not present, and that's very important when you think about the function of those three organs. Okay, so we've brought glucose into the cell, we're making ATP, and I'm going to go ahead and move to the next slide. As we're making that ATP, we're using as much of the glucose as we need, but then we're also going to store the excess glucose, okay? So we're going to make up a bunch of ATP. We got all this glucose left over. We're going to store it, so we need to hook all those glucose molecules together, remember like we talked about a while ago, and form a polymer. Polymer, that's a big word that doesn't really mean anything so complicated. Poly means many, and mer is unit, okay? So we're making many unit structures, okay? So we're taking something, instead of having all those monosaccharides, we're hooking them all together to form that long chain glycogen so that we can store those glucose molecules better. Okay, now we also are not going to use all of that to make glycogen. Some of that glucose is going to be taken into the adipocytes, fat cells of the body. And once it's taken into the adipocytes, it can be used to make more fats, more lipids within that cell, because remember, the glycerol or the glucose can be converted to glycerol, and glycerol is a component of glycerol is a component of fat, because it's fatty acids and glycerols that make fats and make lipids. Okay, I think that's all I wanted to tell you on that one. So let's move to the next one. All right, now this is the mechanism that shows how glucose levels are, or how blood glucose levels are regulated. So if you take the two hormones we've just talked about, the insulin and the glucagon, what's going to happen? Well, let's look at this and see. First of all, can you tell me what type of mechanism this is? Oh, I bet you can. It is a negative feedback mechanism, right? You've seen this before, right? You remember what a negative feedback mechanism is? It's when you make a product or you cause a change to happen until you get to the set point that the body likes, and then the change stops happening and goes back down around the circle there, up the little diagram, until that change occurs again. Just like the thermostat in your house, right? If your house is too cold, your heater clicks on until your house heats up enough that the thermostat recognizes that it's warm enough, so the thermostat shuts off. Well, the same thing is going on here with regulation of blood glucose. Remember, our bodies like to maintain homeostasis. So we know that already, right? Homeostasis. So normal blood glucose levels, normal blood glucose homeostasis. This says between 70 milligrams per, or about 90 milligrams per 100 mils. The easy way to think about this, that's not necessarily the way that we normally see it, is somewhere between 70 to 110 are normal blood glucose levels. Okay, so let's say that blood glucose levels rise, maybe because we just had a big meal. All right, so blood glucose levels rise, and that is going to stimulate the pancreas to release which hormone? Insulin. Yeah, you knew that. All right, so blood glucose levels rise, pancreas secretes insulin. Insulin is going to stimulate the liver to convert glucose to glycogen and store it. It's going to stimulate body cells to take glucose in, make ATP. So blood sugar levels start to fall until we get back to homeostasis. Now let's say that blood sugar levels have fallen. It's been a long time since we've eaten, or we're asleep, or we're exercising and using up glucose quickly to make ATP. So the pancreas is then going to be stimulated to release glucagon. Glucagon then is going to stimulate the liver to take glycogen, break it down to glucose. Glucose gets into the bloodstream, blood sugar levels rise until we get back to homeostasis. So your normal feedback or your normal negative feedback mechanism works with the pancreas to release either insulin or glucagon to keep those levels at the homeostatic ranges. Okay, so now let's talk about what happens when those levels are not where we want them to be, not where our body likes it to be. The most common condition is diabetes mellitus. This big word is what you normally hear just simply called diabetes, but that doesn't describe the actual diabetes that is a result of insulin problems. Okay? You have to have the diabetes mellitus to use the entire term, because there are different types of diabetes. There's gestational diabetes, and there's also diabetes insipidus, and those are very different from diabetes mellitus type 1 and 2. So let's look at the roots of these words. Diabetes means to siphon. You know what it is to siphon, right? You're taking a liquid from one place and moving it to another. You siphon gas from a car, perhaps, or you siphon the water from your fish tank when you're wanting to clean it. So this is siphoning water from the body into the toilet. Okay, so we're creating a lot of urine. Now, mellitus is an interesting term because it means sweet, to taste like honey. So we've got both Greek and Latin. We have the Greek word, diabetes for siphon, and mellitus is a Latin word for sweet like honey. Now, urine, sweet like honey, are you curious? I was, so I had to go out and look it up and see who actually found out that there is a sweet taste to urine in the case of diabetes, and that was in 1679. A physician named Thomas Willis, Thomas Willis actually tasted the urine of a person with diabetes and described it as wonderfully sweet, tasting like honey. How would you like that to be your job? Okay, so let's get serious here. So our kidneys, let's back up to what happens here. This condition, diabetes mellitus, results from hyposecretion of insulin, not enough insulin being secreted, or hypoactivity of insulin, not enough action. The insulin is not causing the cells to take up blood sugar or take glucose in. Okay, so when you're not taking glucose in, your cells are basically starving, okay, because glucose is what's used to create the ATP that gives our body energy to do everything that we do. So it's just like your car's not going to run without gas, your body's not going to run without ATP, and we've got to have something to make that ATP from. Okay, so the glucose is not being taken into the cells are starving, and the kidneys start to secrete excess water. It does that because once the blood sugar levels rise and that is being filtered through the kidneys, a lot of sugar, of course, is being filtered through. So osmosis is going to cause more water to be filtered through as well. Remember, just like water follows salt, well, the same thing is going to occur because the body wants to try to get to normal balance, right? So the osmotic pressure of having all of that excess sugar filtering through is going to cause more water to be filtered through as well. So we excrete more urine. Well, as we're excreting more urine, we're taking water from the body. Our body becomes thirsty. So we have excess urine, excess thirst. This is polyuria, polydipsia. And then the third of our signs here is polyphagia. Phage, P-H-A-G, means to eat. So excess hunger. Remember I said when our body's cells are not getting glucose, they're starving. Well, that sends signals to the brain, I'm starving. So you become hungry. You want to eat more in an attempt to get more glucose into the body. And, of course, it's simply going to be urinated out. So the three cardinal signs, the three polys: polyuria, excess urine output; polydipsia, excess thirst; and polyphagia, excess hunger. Okay, so let's talk just a little bit more about the types of diabetes. I mentioned that there are two types of diabetes mellitus. Diabetes type 1 is caused by hyposecretion of insulin. Not enough insulin being secreted. This is typically caused by the beta cells being destroyed in the pancreas. So not enough insulin is being made. This was formerly called juvenile diabetes because typically it occurs in people under the age of 40. Their bodies are usually very thin. Their symptoms often appear very suddenly as well. And this is also sometimes called insulin-dependent diabetes, okay, because the body's not making enough insulin. The insulin-producing beta cells are being destroyed, so you have to typically take insulin. A lot of times this is injected. There are new forms of insulin that can be used as a nasal spray. So anyway, your body's not getting the glucose that it needs, or not getting the insulin that it needs, so your body cells are starving. Now, type 2 is non-insulin dependent diabetes, and this is also sometimes called adult onset diabetes mellitus. So this happens in people that are overweight most of the time, over the age of 40. Symptoms may appear very slowly, and this can typically be treated by diet and exercise, and a lot of times the type 2 diabetes goes away with diet and exercise. Now, this happens, when this happens, your body is making plenty of insulin. But what happens in some cases is that the insulin is not binding to the receptors, so the receptors are ignoring the insulin, or the receptors may be destroyed, so there's not enough of those insulin receptors. If there's not enough insulin receptors, or the insulin is not binding the receptors, those glucose transporters are remaining down in the cell, in the cytoplasm, rather than being embedded in the membrane to bring all that glucose in. Okay, so that's type 1 and type 2. I mentioned earlier gestational diabetes. Gestational, what does that mean? Gestational, someone that's pregnant. Gestation, the period of pregnancy. So this is when the pregnancy hormones interfere with glucose action, or with insulin action, Gestational diabetes, the pregnancy hormones interfere with the action of the insulin, and of course that's going to prevent it from binding to the receptors, and once again you're not getting enough of the glucose into the cell. And the third type is not on this slide, but it is diabetes insipidus. Insipidus. That means no taste. Once again, not the job you'd want to have, huh? Okay, this is caused by a failure of the kidneys to respond to antidiuretic hormone, ADH, or by a lack of ADH, antidiuretic hormone. Remember what antidiuretic hormone does? Causes the body to conserve water, right? Antidiuretic. A diuretic makes you produce more urine, so an antidiuretic produces or causes you to produce less urine, so your body's conserving water. So if antidiuretic hormone is either not being secreted or your body's not responding to it, then of course there's going to be a lot of urine produced. This can be caused by head injury, certain infections, tumors, certain drugs, so there's several different things that can bring this on. Okay, let's see. Well, I've covered everything, so I guess we're done with this one, and I'll talk to you again soon.